Yeah, that was good.
Excellent. Take it away.
You're looking at the right screen. Yeah.
Yes.
Awesome. Thank you. Okay. Yeah. I just wanted to quickly mention that I am at the
And we are frozen.
Mm-hmm. Might have to move on and come back.
Okay, let's move to Jackson Lab. Maheshwar Thapa. Shrikaar, you're back. You're muted.
Sorry about that. I didn't realize what just happened there.
Okay, let's give it another try.
Yes. Yes. I want to talk to you about some insights that we got from plant lipid metabolism using stable isotopes and high resolution mass spectrometry. And like I said, this work is part of the U01 awarded to Doug, Jamie, and Brad. Doug at Vanderbilt, Jamie at Vanderbilt, Doug at USDA, and Danforth Center, and Brad Evans, who runs the core here. We developed a software tool as part of this work, simple, along with a few others that Jamie will talk about in later presentation today. And I also want to acknowledge and thank the Metabolomics Workbench team who recently integrated Simple into Metabolomics Workbench, especially to Owen and Shrikaar. Thank you. So understanding complex metabolism requires comprehensive dynamic analysis. And in the Allen and Young labs, we use stable isotopes such as 13C glucose to and mass spectrometry to trace the movement of carbon in order to capture the dynamic aspects of active metabolism. People in this audience should be familiar with and can appreciate that HRMS has enabled us to distinguish the minor mass charge differences between isotopic labeled versions of carbon of fatty acids, and also the desaturation that can otherwise be indistinguishable if you're using nominal resolution. And since the talk is about lipid metabolism, I wanted to mention this unique aspect of HRMS that you can gain from lipids. However, when you label with 13C, the longer you label and you look at the mass spectra that you obtain from labeled samples, the longer you label, you end up getting more and more isotopic versions of spectra that can be tedious to integrate if done manually, right? And are also error prone. So we developed a software tool, Simple, that can automate this process and focus more on trying to get at biological aspects of this data instead of worrying about your data itself. Right, as an example, I demonstrate here the use of our tool in understanding plant lipid metabolism using an oilseed model, Camelina sativa, that is labeled with 13C, and then we perform LCMS and use our tool Simple to extract the isotopic labels and analyze the data. Right, we can use mass spectrometric data collected both at MS1 to provide information on label enrichment here at the class level. If I, for example, here I'm showing PC364, and also the MS2 data, where you can get information on the fatty acid labeling within each of the isotopologues or the labeled versions from the MS1, which also contain the isotopologues, right? Fatty acid isotopologues. So quickly with our tool, what you can do is perform identify these isotopologues and perform statistical analysis, such as k-means clustering. Here I'm showing how we take advantage of labeling over time to cluster compounds into whether they are fast or slow, and take advantage of the speed of labeling to assemble biological networks. And you can do that at MS1 and also at MS2 look at the fatty acid labeling also over time in each of the individual lipid classes. Right, as quickly, without going into the details of the data, I wanted to provide the biological insight that we gained, which is the question that we're trying to answer is, oil seeds produce an enormous amount of polyunsaturated fatty acids. Over 60% of the entire fatty acids are polyunsaturated fatty acids. But if fatty acid synthesis in plants produces 16O, 18O, or 18-1 that is unsaturated and monounsaturated fatty acids, how do you end up getting so much polyunsaturated, right? Traditional understanding of lipid metabolism involves fatty acid synthesis and its incorporation of these acyl chains into triacylglycerols through Kennedy pathway. What we found is that PCs, phosphatidylcholines, label much faster from our labeling data. PCs label much faster compared to diacylglycerols, which are part of the Kennedy pathway, suggesting that fatty acids needs to be incorporated into PC first, and PC is the site for desaturation using our MS2 data, with the 18-1s quickly converting into 18-2s and 18-3. We identified that these desaturated versions get incorporated into DACs before going into triacylglycerols and forming oil bodies. Right, so this is not unknown. However, this is one of the quickly assembled pathways using isotopic labeling with our tools. So these tools make isotopic labeling data analysis more accessible to researchers, and it's also publicly available right now. Thank you.